Hello everyone. So in this lecture, we will be talking about the metabolic pathway glycolysis. Basically, the glycolysis is the pathway that is responsible for the breakdown of glucose molecule into pyruvate molecules and also in the generation of energy in the form of ATPs. Now, when we talk about glucose, so glucose basically arises in the blood as a result of breakdown of complex polysaccharides. So when we consume carbohydrates through diets, they mostly comprise of the polysaccharides. These polysaccharides are acted upon by different enzymes, and these are broken down subsequently into glucose. And this glucose is the glucose which then undergoes glycolytic pathway to form pyruvate. Now, the enzymes that are present in the glycolysis pathway, they are present in the cytosol. So all the enzymes, they are sequestered in this organelle. Now let's see that what exactly is happening in the glycolytic pathway. So when we talk about glycolysis, the glucose molecule, which is a 6-carbon molecule, it is broken down into 3-carbon molecules. So basically 2 pyruvates of 3 carbon each are formed. And the energy that is released in this breakdown, it is utilized to synthesize ATPs. And that is what we are going to see in this lecture, that how exactly these ATPs are being formed and on which reactions they are being formed. Now, as I mentioned just now, that glycolysis takes place in the cytosol because all the enzymes that are responsible for carrying out this pathway are present there. What happens is that if there are aerobic conditions present inside the cell, then this pyruvate that is converted from the glucose, it enters into another pathway called citric acid cycle, and there is a generation of more energy. Now, this happens when oxygen is present. Now, what happens if oxygen is absent, which means that anaerobic conditions are existing inside the cell? In those conditions, lactate will be formed. Or if alcoholic fermentation is taking place, then ethanol will be formed. So these are the basically 3 fates of pyruvate from the glycolytic process. Now, this glycolysis is also the major pathway for the ATP synthesis. This is the first pathway which is basically producing the ATPs. And the glycolytic pathway is having 10 steps, which we are going to discuss now. So let's move on and try to discuss all these steps one by one. So if I have to divide glycolysis, because as I told you that there are 10 reactions in glycolysis. So if I divide these 10 reactions into different phases to make you understand it better, there are 3 phases. So out of these 10 reactions, there are 3 phases. The first phase is known as the energy consuming phase. The second phase is known as the splitting phase. And the third phase is known as the energy payoff phase. So basically, let me give you an example. Let's suppose that you invested 10 rupees or $10 in a bank. After a year, you are getting back 20 rupees or $20, right? What does that mean? This means that you are having a net profit of 10 rupees or $10. Now, if you would have not invested 10 rupees, how would you have got 20 back and 10 net profit? So that is what's happening in the glycolytic process. So in the first phase, you have to provide some energy for the reactions to take place so that in the last phase, you can get more energy back so that you are having a net gain of energy. So that is what exactly is happening inside the glycolytic process. Now let's see all the reactions one by one. First, we will talk about the energy consuming phase. So out of the total 10 reactions of glycolysis, the first 5 reactions, or I would rather say the 4 reactions, first 4 reactions are a part of the energy consuming phase where we are providing energy for the reactions to take place. So the first reaction is when glucose is converted to glucose 6-phosphate. So this is the glucose, which is the 6-carbon molecule. On this glucose molecule, an enzyme hexokinase acts in the presence of magnesium ion, And it also uses the energy of an ATP, and this glucose is converted to glucose 6 phosphate molecule, right? So this first step of glycolysis is basically involving the transfer of a phosphoryl group from the ATP molecule to the glucose to form glucose 6 phosphate. So phosphoryl group from the ATP molecule is removed by this hexokinase and it is added on the 6th position of glucose like that. And this ATP, after it has lost its phosphate group, gets converted into ADP. So that is why ATP energy is being consumed here, because that ATP is being utilized for providing the phosphate group to the glucose to convert it into glucose 6 phosphate. Now, talking about the specific enzyme hexokinase, this is a very important enzyme. So this is a kinase enzyme, and kinases generally are the enzymes that are responsible for transferring the phosphoryl groups between ATPs and the metabolite molecule. And here the metabolite molecule is the glucose. So phosphoryl group from ATP is being transferred to glucose to convert it into glucose 6-phosphate. Also, make sure that you remember that magnesium ion here is important for this reaction to take place because hexokinase is not going to act without its presence. So this was about the first reaction. And before we move on, I told you that there are total 10 reactions in the glycolytic pathway. Out of this 10 reactions, 3 reactions are irreversible. 3 reactions are irreversible and the remaining 7 are reversible reactions. So the 3 reactions that are irreversible are the reaction number 1, 3, and 10. So the first reaction we have just discussed, this one, and this reaction is irreversible, which means that this hexokinase is going to convert glucose into glucose 6-phosphate, but it does not hold the capability of converting glucose 6-phosphate back into glucose. So this is an irreversible reaction. Also, remember that it is consuming energy of an ATP. So this was about the first reaction. Now let's move on to the next reaction. So after the glucose 6-phosphate has been formed, sorry, this is the second one. I have to go to this one. So the second reaction is when an enzyme called isomerase acts on the glucose 6-phosphate and converts it into fructose 6-phosphate. So isomerase is an enzyme that is going to perform isomerization. Now, glucose and fructose, they are isomers of each other. So this isomerase enzyme is going to act on glucose 6-phosphate and convert it into its isomer, that is fructose 6-phosphate. So this basically is involving the isomerization of an aldose to a ketose. Now, this is an aldose and this is a ketose, right? So this was the second reaction, and this reaction is a reversible reaction. Moving on, the third reaction of the energy consumption phase is that this fructose 6-phosphate that has been formed in the second step, it is converted into fructose 1,6-bisphosphate. Now, how is this happening? Let's see. So this fructose 6-phosphate is having a phosphate group on the 6th position. Here, an ATP will come again, and a kinase enzyme specifically called phosphofructokinase is going to act on this, and it's going to remove the phosphoryl group from the ATP molecule, and it is going to attach it to the first position of the fructose 6-phosphate as well, and subsequently, the molecule fructose 1,6-bisphosphate will be formed. So as I previously mentioned, that kinases are basically the enzymes that are responsible for transferring the phosphoryl group from the ATP to the metabolite molecules. Here, the metabolite molecule is fructose 6-phosphate. The phosphoryl group is being removed by the phosphofructokinase from the ATP, and it is getting attached on the first carbon atom. So after this ATP has lost its phosphoryl group, ADP is being formed. Now, this reaction is the third reaction, and this is also an irreversible step. Also, this enzyme requires magnesium ion for its functioning. Now, make sure to remember that the reaction number 3, which is being catalyzed by the phosphofructokinase, is the committed step. It is the first committed step of the glycolytic pathway, which means that if this step takes place, then the pathway has the tendency to move towards the end, right? Okay, so this was the third reaction. Now let's move on to the fourth reaction. So after fructose 1,6-bisphosphate has been formed, now comes the next step where an aldolase enzyme is going to act on this, and it is going to split this 6-carbon molecule into two 3-carbon molecules. Now you might have observed that I was explaining the structures in their Haworth projection. Now I have formed the chain form, that is the Fisher projection, so that we are able to understand the chain formation, and we are able to understand that how the 6-carbon molecule is being broken down into two 3-carbon molecules. So this is a 6-carbon molecule. Aldolase is acting on it, and it's going to cleave this molecule into two individual components having 3 carbons each. So what are these two 3-carbon molecules? So one of the molecules is known as the DHAP, that is dihydroxyacetone phosphate, and the second molecule is known as the glyceraldehyde 3-phosphate. So these two molecules are again isomers of each other. Now, so what happens is that this glyceraldehyde 3-phosphate molecule has the tendency to move forward towards the payoff phase of the glycolytic pathway, that is the next steps. But this DHAP does not have the capability of moving forward into the glycolytic pathway. In order for this DHAP to move forward in the glycolytic pathway, it has to convert into G3P, into the glyceraldehyde 3-phosphate, right? And how does that happen? It happens via an enzyme called triose phosphate isomerase. So let me explain it again. This fructose 1,6-bisphosphate, it was acted upon by aldolase enzyme and was converted into two 3-carbon molecules. One is DHAP and one is G3P or GAP. This GAP has the tendency to move forward into the glycolytic pathway, but DHAP does not have it. For this molecule to move forward, it has to first get converted into glyceraldehyde 3-phosphate. Now, how is this happening? This is happening in the fifth reaction, which is the interconversion of this glyceraldehyde 3-phosphate, this dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. And this is acted upon by triose phosphate isomerase. So this triose phosphate isomerase is basically going to perform isomerization where DHAP will be converted into its isomeric form, that is glyceraldehyde 3-phosphate. So after the fifth reaction, we are now left with two molecules of glyceraldehyde 3-phosphate. So whatever I'm going to explain now further, it's going to happen twice, because we have two glyceraldehyde 3-phosphate molecules, and all the reactions that are going to happen now in the glycolytic process, they are going to happen twice. So whatever will be formed will be twice in number. Now let's see the sixth reaction, that is the energy payoff phase. Now in the first 5 reactions, we saw that we have been providing energy, and basically the energy was being provided in the form of ATPs, especially in the reaction number 1 and the reaction number 3. Now comes the energy payoff phase. Let's see. So in the reaction number 6, the glyceraldehyde 3-phosphate that was formed, it is converted into 1,3-bisphosphoglycerate. And how is this being formed? This is being formed with the help of an enzyme called glyceraldehyde 3-phosphate dehydrogenase. So what it is doing, it is basically removing the hydrogen atom from the glyceraldehyde 3-phosphate and attaching it to the NAD molecule, and a phosphate group is being attached on the position number 1 as well. And from where this phosphate is coming, it is coming from the inorganic phosphate. So this was the reaction number 6. This is a very important step because this involves the formation of NADH, which is a high energy molecule. And also this 1,3-bisphosphoglycerate, which has been formed, this is also a high energy compound. Now, when there are aerobic conditions present inside the cell, this NADH so formed can enter into the electron transport chain to produce more energy. So that is why we are referring to this phase as energy payoff phase, because it is producing energy now. Now let's move on to the next reaction, the reaction number 7. So in the reaction number 7, the 1,3-bisphosphoglycerate that was being formed, it gets converted into 3-phosphoglycerate. Let's see how it is happening. So you see in 1,3-bisphosphoglycerate, there are two phosphate groups present, one on the position number 1 and one on the position number 3. So an enzyme called phosphoglycerate kinase will act on this molecule, and it is going to remove the phosphate group from the position number 1, and it is going to attach this phosphate to the ADP molecule to convert it into ATP. So the ADP was basically having two phosphate molecules. Now one phosphate molecule has been removed from the position number 1 and incorporated into the ADP molecule to convert it into triphosphate. So you see why we are calling it energy payoff phase, because ATP is also being formed now. You see that energy is being released now in this phase. Okay, so this was the reaction number 7. And this reaction is also an example of substrate level phosphorylation. Okay, moving on to the next step, the next reaction, that is reaction number 8, where this 3-phosphoglycerate is converted to 2-phosphoglycerate. Now, the main difference between these two molecules is the position of the phosphate group. So what happens is that in the 3-phosphoglycerate molecule, the phosphate is attached to the position number 3, the carbon number 3. But now the mutase enzyme is going to act on it, and it is going to transfer this phosphate group to the position number 2, the carbon number 2 basically. So this mutase is doing this thing. So this is an isomerization reaction, you can say. Okay, moving on to the next reaction, the reaction number 9. Here, the 2-phosphoglycerate is converted to phosphoenol pyruvate. Now, this phosphoenol pyruvate is also a high energy compound, and this molecule is generated with the help of an enzyme called enolase, which also requires magnesium ion or manganese ion to work. So, for the activity of enolase, these enzymes are, these ions are important. So now let's see what's happening here. So if you see at the structure of the 2-phosphoglycerate, you can see that an OH group is present here. So now when enolase is acting on it, it's just removing this OH group from here and hydrogen atom from here, and double bond is being introduced here, and water is being released in such a fashion. Now this molecule that has so been formed, this is known as the phosphoenol pyruvate, which is a high energy compound. Moving on to the last reaction. So the last reaction, it is again an irreversible reaction. So you see that when we were doing the energy consumption phase, we saw two irreversible reactions, the reaction number 1 and 3. After that, whatever we have been studying, everything was reversible. But now this reaction number 10, this is irreversible. This is being catalyzed by an enzyme called pyruvate kinase. Again, what is a kinase? Kinase is basically the molecule or the enzyme which is responsible for transferring the phosphoryl group between ATPs and the metabolites. So here the phosphoenol pyruvate, which is having a phosphate group here at the position number 2, the phosphoenol pyruvates, this phosphate group is going to be removed by this pyruvate kinase, because this is a kinase, right? It's going to do something with the phosphoryl group. It's going to remove this phosphate group from the position number 2, and it is going to incorporate it into the ADP molecule to subsequently form ATP. So this is again a very important example of the reason why this phase is known as the energy payoff phase. Now let's sum it up all and see that what exactly is being formed at the end of this pathway. So you see in the reaction number 1, 1 ATP is being consumed. Then in the reaction number 2, again 1 ATP is being consumed. In the reaction number 6, 1 NADH molecule is being released, right? In the reaction number 7, 1 ATP is being released. And in the reaction number 10, again, 1 ATP is being released, right? So in total, we have used 2 ATPs in the energy consuming phase, and we have released 4 ATPs in the energy releasing phase. So in the energy consuming phase, we used 2 ATPs, and in the energy releasing phase, we released 4 ATPs. Why there are 4 ATPs? Because you see that after the 5th reaction, there were 2 glyceraldehyde 3-phosphate molecules, and all these reactions, they are happening twice. So because with one glyceraldehyde 3-phosphate molecule, 1 ATP is being formed. Simultaneously, with the second G3P molecule, the second ATP is also being formed. So in total, whatever is happening twice. So there are 4 ATPs which are being released. So there is a net gain of 2 ATPs. So that is what the balance sheet for the glycolysis says. In the next lecture, we will be talking about the Krebs cycle. Thank you.